And just to remind everyone, we will stop recording at the end of the main presentation before the Q&A. All right, well, welcome to today's Consciousness Club. It's a real pleasure today to be hosting Rafi Malach, who's a professor of brain research at the Department of Neurobiology at the Weizmann Institute of Science in Israel. And Rafi's research group aims to uncover processes that lead to the emergence of conscious sensory perception in the human brain. And I think uniquely among many labs pursuing this, he combines both non-invasive techniques with also invasive electrophysiological recordings in patients. So Rafi did his PhD at UC Berkeley and then a postdoc at MIT. Since then, he's made many contributions to the understanding of perceptual recognition, perceptual awareness, the hierarchy of visual processing. He was the 2022 recipient of the Emmet Prize in Life Sciences, often referred to as Israel's Nobel Prize, and he's a CFAR Tannenbaum Fellow in the Brain, Mind, and Consciousness Program. And as you'll see in today's talk, Rafi's more recently been doing some really interesting, cool stuff, looking at the relational structure in conscious experience and how that might be supported by the human brain. So we're delighted to have you, Rafi, to give today's Consciousness Club, and over to you. The floor is yours.
Great. Thanks, Steve. And I'm really glad for the opportunity to speak in this distinguished seminar. And thanks for the invitation. Before I start, I would like to say that a substantial part of the stuff that I'll be showing is still unpublished. So I'm really appreciate feedback, comments, questions, criticism, and so forth. Feel free, if it doesn't occur in this forum, to write to me, and I'll be glad to hear comments. So let's start. My sort of starting point is what I call a perspective, the localist perspective. It was originally proposed by Semir Zeki, and the idea is, and others, and the idea is that each local region in the human cerebral cortex underlies or informs about a specific category of phenomenal experience. So when you move from one region to another of part of the cortex, each part underlies a different category of phenomenal experience. Of course, in the back, we have contents related to vision, on the sides, maybe to sound and so forth. So this is the basic idea, that we are linking content, categories of content of perceptual phenomenal experience to different regions, localized regions in the cerebral cortex. Now, a conundrum immediately springs out from this perspective. And I will illustrate this conundrum with this example of two visual areas, well studied, that are actually neighboring each other. One is, you see it here in the back, it's called the lateral occipital complex. This is a visual area that is associated with perception of objects and shapes. And right near it, not far away, about 10, maybe 10 centimeters away, there is another area called MTV5, which is associated with the experience of motion. Now, it's not that every pattern that ever existed or occurred in the lateral occipital complex was always associated with object, and every pattern of activity that occurred in MTV5 was always associated with vision, with motion. And the thing is, it's not only when you are stimulating due to feedback from the lower parts of the visual system, when you are stimulating the person with visual moving targets or with shapes, you can get the same kind of phenomenal experience by recall, memory activation, sometimes in hallucinations, sometimes by electrical stimulation. Directly electrically stimulating these regions will elicit always motion here and always shape and object or faces, in this case here. So what is special about this separate region that is confining the category of phenomenal experience in one case to shapes, and the other case to motion. And my suggestion, the general framework that I would like to propose, is that the source of this specialization or category association is related to the fact that what really determines the phenomenal experience is the relation of this phenomenal experience to other experiences. For example, and this idea related to structuralism is a very old idea. It was actually suggested many years ago by in the linguistic domain by Ferdinand de Saussure. And the idea is very simple. You see this face appears to you as this face, not because what it is, but because it is close, similar to these faces and far away from these faces. The matrix of relationships between this face and other faces, this is what determines the phenomenal content of this face. Now, the question is, of course, how do you implement or how this relates to neuronal representations? And I apologize here for those of you who are familiar with this concept, but it is very important that we really are on the same page in terms of the concept I'm proposing and the terminology, because this will be the basis for my entire presentation. So the way to think about relational structures in the neuronal domain is to contrast them with the more dominant concept of population vector. So let's start with population vector. A population vector is the idea that the face of Bruce Willis is defined by a population of neurons that are activated by the face of Bruce Willis. And these neurons, as a group, as a collective, define a profile of activation. So, for example, this is actual data. Looking at this face of Bruce Willis, you get a group of neurons recorded by Itzik Norman and Rothenborn that some of them are responding very high to Bruce Willis and some of them respond very low. And together they form a profile. You can sort of metaphorically think about it as if there is a barcode on the cortex. Some neurons activated strongly, some not. And together, this barcode pattern of activation defines the percept of Bruce Willis. We can sort of present this in a vectorial notation by defining a space. This case, it's just a caricature space made up of only two neurons. And in that case, this profile or this pattern of activation population vector is actually a vector made up of two dimensions, activity in this case, one neuron, and activity of another one. Together, the joint activity defines a point in this vector, and this is the population vector. Of course, in real brains, there are millions of dimensions in this space, but together they will define a point, and this point is actually the neuronal coding according to the population vector idea. This is the neuronal coding that underlies the appearance of Bruce Willis. I hope this is clear. Really, a good metaphor is a barcode. Bruce Willis is associated with barcode, and another person will be associated with another barcode, a different profile of activation, and so forth. Now, contrast this with the idea of relational coding. Here, the reason Bruce Willis appears as Bruce Willis has nothing to do with the specific profile of activation. It is not a consequence of the specific population vector associated with Bruce Willis. What determines the appearance of Bruce Willis, according to the relation coding idea, is the set of distances. In other words, very simply, the similarities of the profiles. You see here, the neurons are exactly ordered in the same way in all these profiles. But you see here the responses, in this case, to Bruce Willis, arranged nicely in a descending order. And in this case, the response of the very same neurons arranged in the very same order to Uma Thurman. And the similarity or the correlation of the vectors, if you want to speak precisely, is determined what we call the distance between Bruce Willis and Uma Thurman. And the set of distances or set of similarities and dissimilarities of Bruce Willis to other images in the same category will determine how Bruce Willis looks like. This is a very important conceptual change that you need to make. The pattern could be completely different, has a completely different profile. If the similarity distances will remain the same, you'll still see Bruce Willis as it is. In the vector notation, we are saying that what really determines the appearance of Bruce Willis is not the vector, but the angles between the vectors pointing to Bruce Willis and the vectors pointing to other images. So we are moved from the absolute coding of each phenomenal content by a specific vector, a specific barcode, to a matrix of relationships in which what determines the phenomenal content is the relation to other content. And of course, what will determine Uma Thurman here is the distance to Bruce Willis and to the other object as well. Now we can ask, what is the set of distances and similarities and dissimilarities between each image and all other images? And in that case, we can form a matrix in which we simply ask for every pair of images, so example, these two pairs, what is the similarity between their population vectors? How correlated they are? The set of all possible, the matrix of all possible distances between all possible pairs of contents within a category is the well-known representational dissimilarity matrix. And basically, you can think about it as the kind of the barcode of the structure. We call this a structure. And this structure is sort of depicted or uncovered by looking at the representational dissimilarity matrix. We uncover the structure, the relational structure, by looking at all the possible distances between all possible images. So the visual percept is coded by the relationship between one pattern and all other patterns within a structure. Now, it is crucial to understand two things. The idea about the local relational structure is that as you move from one cortical region to another cortical region, you change the relational structure. And I illustrate this, and this change in relational structure in the set of all similarities and distances is actually defining the category that is coded by this localized region. And I want to illustrate this by considering two areas. These are, of course, still in the proposal idea, but I think the evidence is supporting it quite robustly. We will contrast the fusiform face area. This is a high order visual area that is selective for faces, with the medial temporal lobe, particularly the hippocampus, where you find neurons that Itzhak Fried studied that they call concept cells. We think that in the fusiform face area, what is really the phenomenal content that you are experiencing is the appearance of the image. You're simply treating the image as a picture, and you ask yourself how this picture look like. So you see these two pictures, and obviously that by this phenomenal category, these two pictures are very different from each other, because this picture, which is the front face of Barack Obama, is round, while here it is more convoluted and so forth. So in the pictorial geometry that defines how things appear to us, these two images will be very far from each other because they appear very differently. These two images will appear closer because they have the same shape and so forth. So you see that in this structure, coding for appearance, we have a specific relational distances. In particular, these two images are far and these two images are close. If you now move to the hippocampus, where we think what is coded is not the appearance of Barack Obama, but the identity who is this person, Barack Obama, the president of the United States, then, of course, the relational structure, first of all, changes completely. Now, these two images are, of course, very close to each other because the identity does not change as the face changes its appearance. And these two images will become far away because these two individuals have very different identities. So you can see that how by changing the relational structure, the distances between images, you change the underlying or the expressed phenomenal content, and how this is implemented as you move from one cortical region to the other. So I hope these ideas are all clear now. And basically, I want to show you now experimental results from our group that try to explore different aspects of this idea of relational structure. And what you see here is the basic experimental paradigm. In this presentation, I'll focus only on intracranial recordings. These are, this is a very invasive method that is conducted in clinical patients, epileptic patients, for diagnostic purposes only, of course. And it is very, very different from non-invasive methods like fMRI, EEG, and MEG. In this methodology, the neurosurgeon, in order to search for the epileptic focus, is inserting long electrode. You see here a schematic drawing of such an electrode. These are long electrodes that are inserted into the brain of the patients that contain many, many contacts. The contacts collect activity from groups of neurons and sometimes even from individual neurons. And the neurosurgeon, because they typically don't know where the epileptic focus is, and sometimes there may be many epileptic foci, places many such electrodes all over the brain of the patient, waiting for epileptic attacks to occur, and then they can find with sort of triangulation where the epileptic focus is. Now, in between, the patient walk with this invasive electrode collecting data, sometimes for a week or more, waiting for epileptic attacks to occur. And in the intermission between the clinical procedures, some of them volunteered to conduct visual experiments. And this is what we do. We do a visual experiment on them while we are recording at the same time this activity from the individual contact. As I said, this is a summed activity of small groups of neurons. For those of you who are interested in the actual signals, what we really measure and what we found to be associated with the average neuronal activity of groups of neurons is not the amplitude of the local field potential like we typically do in EEG and MEG recordings, but the power, the amplitude of the tiny fast oscillations that happen in the signal. It's a kind of a buzz, you might think about it, that comes out of the local field potential. And we and others found that it's correlated very nicely with the average activity, spiking activity of the neurons. And the experiment, basically I show you the basic sort of skeleton or framework of the experiment. All other experiments will be variations on this theme. This is data that was collected by Shani Grossman. And the experiment is actually very simple. The patient faces a computer screen. We are flashing pictures of different categories of visual pictures, faces, places, textures, and so forth. In this experiment, for 250 milliseconds with intervening periods of 700 to 1,000 milliseconds. Occasionally, a picture will appear, repeat, and then the patient will have to press a button. This is the basic framework. All experiments I'll be talking today will be around this framework with some variation that I'll mention. We collect these recordings from all over the brain, but I'll point to specific cortical regions according to the results that I'll be showing. So I hope this general framework is clear. Very simple experiment. This is important in these patients to have the experiment easy understood and performed. And basically, they're watching pictures of different images. And we are looking the activity in different cortical areas to these regions. So the first question, if you think about it, the idea that I propose that really what determines the phenomenal appearance of faces in the face area, if it's true, then there is a very simple prediction that comes of it, and this is very simply that those faces that generate similar population vectors, in other words, that have short distances between them in the neuronal domain, should also appear to us to be similar to each other. I hope this is clear. So we would predict that those faces that generate patterns that are close to each other in terms of the profile will appear more similar than faces that generate population vectors that are very distinct from each other. And this prediction was tested by Ido Davidescu, who did a very simple experiment. On the one hand, he presented in a typical experiment like I showed you before, he presented these patients with different images of faces, unfamiliar faces, one face at a time, and recorded the activation patterns, the vectors, population vectors for each face. And in parallel, this was done in the United States. In parallel, in Israel, in the Weizmann Institute, he printed those faces on cards and asked students to simply bring those cards together at short distance if they are close and similar to each other, and put them at far distance if they are very different from each other. Here, for example, are two faces that the student decided were very similar to each other, and here these are two faces that the student decided were very far from each other. And then what you see in this scatter plot is based on the recordings done in the face area that I mentioned, high order face area. And what he plotted, Ido plotted here on the y-axis, the neuronal distance between the activation to these faces. In other words, how far away or close they were in the terms of the population vector, how similar they were. And on this axis, he plotted the perceptual distance. Now, the prediction, if the relational coding is really inducing the feeling of similarity and distance, then we will predict that there will be a strong correlation between the perceptual distance, in other words, the appearance of images as being similar or not similar, and the neuronal distance, the activation of these faces to similar patterns or faraway patterns. And indeed, he found a nice significant correlation. And what's even more reassuring is that if we focus the analysis only on those contacts that were more consistent, the relationship between the perceptual distance and neuronal distance even increased. So this is a nice sort of fulfillment of the prediction that the relational distances should underlie the similarity appearance of images. Interestingly, if we now moved and instead of looking at the patterns that are generated in the high order face selective regions, Ido looked now in early visual cortex, areas V1 and V2. These are the areas that are receiving the input from the eye before it progresses to a high order area. Here, first of all, we find two things. First, the relational structure completely changed, and we see that because this nice correlation that we saw before disappeared. And secondly, we don't see any relationship between the neuronal distances in early visual cortex and the phenomenal experience of face similarity. So at least if you accept the premise or the suggestion that the relational structure is underlying phenomenal content, then this shows that early visual cortex, there might be underlying different aspects of phenomenal content. They definitely don't code for the similarity or the appearance of faces. The next question I would like to address is a study led by Advas Shoham from the group of Galit Yovel in Tel Aviv University, together with Rotem Bodei Dvir and Itai Yaron. And this deals with a question that is of great interest to theories of consciousness that you might know. And this is, what is the relationship between visual activation in the back of the brain, in the classic visual cortex, occipitotemporal areas, and so forth, and the frontal cortex? Areas that are activated visually in the front part of the brain. And the experiment is again a variation on the same experiment I mentioned before, intracranial recording in patients, many places in the brain. This is actually work done with Itzhak Norman in my lab. And basically the patient in this case is looking for images of familiar faces and familiar or famous places. So famous politicians, actors like you saw, and so forth. Notice that the presentation time of each image is much longer. It is 1.5 seconds. And another thing is that the patient didn't have any specific task that they needed to conduct when they looked at this image. After they were exposed to these famous places and famous faces, they were in a separate session, and I will not discuss this result. They were asked to recall the images that they saw before, and in this case they recalled very well, showing that they were aware and saw the images and so forth. So again, experiment very simple. You look at famous faces, famous places for 1.5 seconds each, while we record responses from all over the brain. And here you see that we found also visual responses in the front of the brain. And what I will do now in talking about the data, I will not cover the entire complexity. I'll focus on 2 regions of interest. One of them is the frontal parietal contacts, which you see here in green. And the other one is high order, again, our high order phase selective regions as an example of high order visual cortex, but we actually see similar results for other visual cortex in the occipitotemporal cortex. So we are now contrasting the relational structure of the frontal lobe with the relational structure of the occipital temporal lobe, in this case, selective for faces. First of all, let's simply look at the activation, because this is an important and debated issue. I'll remind you that there was no specific task that the patient were asked to do. They were simply asked to look at these images carefully for 1.5 seconds. Nevertheless, we see nice activation. We see, in fact, activation all over the cortex. For example, in early visual cortex, very robust activation. We see, of course, very beautiful activation in phase selective contacts. But importantly, we see a very significant and robust activation in the frontoparietal visual electrodes. So frontal lobe definitely responds to visual activation. Actually, it responds at a time course that is not much different from the phase selective responses. So just looking at this activation, we cannot tell very much about the relational structure, of course, of underlying these different activations. We can just note that there is activation both in the occipitotemporal cortex, in the frontal parietal cortex. If you look, we can actually construct a relational structure diagram. I mean, the representation dissimilarity matrix. I remind you, this is simply plotting all the distances between each individual face and each individual place and all other images. And this is the RDM, the representational distances or the relational structure as appears in the phase selective high order visual cortex. And this is what you see in the frontoparietal visual activation. You see very striking difference between them, but it's very difficult to decipher what exactly these two RDMs mean. I mean, what do they underlie in terms of the category of phenomenal concept, or what are they actually coding, what are they specialized for, and so forth. And here Adva took advantage of recent discoveries done in my lab, in other labs as well. And this is the interesting finding that if you look at the relational structure of, for example, face cortex, and compare it to the relational structure that you derive from artificial networks, this is usually done by looking at the top, one before the top layer of each of these artificial networks. How do you get a relational structure from these networks? Very simply, you feed them with stimuli. In this case, what Adva did, she fed the networks with the same images that the patient saw, so there will be famous faces, famous places. You feed these images into the network, you record the activation pattern in this high order layer, the embedding layer, so it's so-called, in these networks, and you again measure how similar or different these patterns, how do they change as you change the images that you play, that you feed into the network, and you map the matrix of relationship in each of these networks that emerge from feeding them with the inputs that you select. So I hope this is clear. You can get, just like you do in the brain, in human patients, you show them different images and you calculate the relational structures. You can do the same thing in artificial networks. You feed them with the inputs and you calculate the distances between the different inputs and get an RDM from it. Okay. Now the important thing that Adva did, and this follows a strategy developed in the lab of Galit Yovel, is that she did this for 4 different artificial networks. The first 2 artificial networks are well-known networks. You might even be familiar with them. The first 2 networks are associated with analysis of visual images. VGG is a classic visual classifier. You feed it with different images and it tells you whether it's Barack Obama or somebody else or whatever. Clip image is a slightly different one. You feed them with different images and it associates these images with a specific text. But still, both of them receive as inputs visual images. Contrast it with 2 other artificial networks that are now gaining enormous popularity, and these are text-related networks. Now, the patient did not see, we did not show the patient text, but you could sort of deduce that when they see Obama, a kind of text that might emerge in their mind will be the name of Obama. So basically, Adva fed clip text with semantic information, the name in this case, of the different famous places and faces. So for example, if there was the Eiffel Tower, she fed the network with the text Eiffel Tower. She also did it with GPT, but here she used even a more sophisticated semantic description. She took the Wikipedia section describing who Barack Obama is, and she fed the entire section into GPT. But again, the bottom line is that you generate a matrix of all the relationship between here, all the images of the famous faces and famous places, and here, the different semantic descriptors of these images. And you generate RDMs from each one of them. And the question now is, of course, how these 4 different RDMs, each one, 2 of them generated by visual networks, and 2 of them are generated by text networks, how do they relate to the different cortical regions from which we got biological RDMs? So let's start with the visual domain with the face selective high order area. And you see here a plot of the correlation between the relational structure in the fusiform face area and the relational structure of these 4 different networks. 2 of them are visual and 2 of them are textual. And here the picture is very simple and actually corresponds to previous studies in our lab and others. You see a very strong correlation between the artificial network, artificial relational structure, and the biological relational structure of the phase selective ventral contacts, but you see very little correlation with the text, and even clip image for some reason didn't show even any correlation. So very striking, clear-cut result, very strong correlation to the visual aspect of the relational structure as revealed by the correlation with the artificial network. Contrast this with what we see when we do the same procedure. I remind you, correlating these networks with the relational structure in the other area when we do it in the frontal lobe. Here the picture changes dramatically, which tells you two things. One of them you could already see just by looking at the RDMs. Moving from the back of the brain to the front completely changes the relational structure. But here we can see something interesting happening. The visual aspects, as defined by the RDM of the visual networks, is reduced substantially compared to the face area. And instead, what really is enhanced and becomes significant are the text-related relational structures. It's as if the frontal contacts, although they respond to the visual images, to the presentation of the visual images, actually correlate with the textual semantic aspects more than they correlate with their visual aspects. So the response is visual, but in fact, if you dig down into the relational structure, you see that it is associated with text, with semantics, and not with vision, at least looking at the relation to artificial networks. Now, I don't think that in this audience I need to elaborate very much, but there was a lot of interest in what's called the adversarial collaboration. That the idea is very elegant, and actually one of the collaborators on this project is a member of Liad Mudrik's lab, who is a major leader in this adversarial collaboration. For those of you who are not familiar, I will say the idea is very elegant, and this is like taking two competing theories, in this case, global workspace and IIT, asking the leaders to propose experiments that will sort of bias or test these theories one against the other, and then actually going and doing the experiments as objectively as they can and seeing which one of the predictions was correct. And this is from the paper describing this adversarial collaboration by Meloni et al. And I actually picked this figure from their paper in Science because there was one prediction that is very intriguing and counterintuitive, done by Stan DeHaan, who is the leader of Global Workspace, and it is depicted here in this figure. And the prediction was about the dynamics of the responses in the frontal lobe when you present a picture for a relatively long time, just like we did, 1500 milliseconds. And the counterintuitive suggestion, prediction coming from Global Workspace theory, was that the onset, you'll get a response in the onset of the response, of the stimulus, and then it will turn silent, and then at the offset of the response, when you stop the stimulus, you should get a reactivation of the original response. Counterintuitive, interesting, and Adva thought that she actually could examine this kind of dynamics, predicted dynamics, by looking into the correlation with artificial networks. How did she do it? She subdivided the time from the onset of the stimulus to the end of the stimulus into time periods, time windows, and for each time window now separately, she did the correlation between the RDM during this time window with the artificial network RDM. So you take different time windows within the biological response, and you correlate each time window now separately with the artificial RDM, which, by the way, don't have dynamics. They just respond in a clockwise manner. So I'll show you first the dynamic correlation between the biological response in the frontal lobe with the visual component as determined by the VGG correlation to the VGG relational structure. What you see, interestingly, is that although on average the visual response was very weak, there was a transient increase in the correlation to the VGG. I remind you, purely visual network. There was a transient increase in correlation in the visual component, but then it declined completely to baseline very rapidly. So there is an initial sort of response to the input that comes from the visual system, but then it declines very rapidly. Contrast this correlation with the visual network with the dynamic correlation to the clip text, which is a semantic network, and again, the correlation to the text RDM a long time in the frontal lobe. And the picture changes completely. You see two things. First of all, the response is more transient and more sustained. This is the correlation to the semantic aspect. Interestingly, following compatible with the prediction of global workspace, it goes down to almost zero, and then at the offset, it jumps to a significant level. So we see here actually confirmation of a counterintuitive prediction proposed by global workspace, but of course, not in the visual domain as proposed by the original formulation, but actually looking at the semantic or textual domain that behaves indeed in different dynamics. And this shows that if you look in the correct relational structure, you might get different dynamics also, not only different cortical regions and so forth. So we see that in fact, what the data is supporting is a more semantic or textual basis for frontal activation compared to the visual one that is more prevalent in the occipitotemporal cortex. The next project I would like to discuss is to ask, how do relational structures change from one brain to another? This is work done in collaboration with Ofer Lipman, Shani Grossman, Doron Friedman, and Yaakov Hel-Or. Shani is from my lab, and Doron, Ofer, and Yaakov are from the Reichman University. And why is it interesting? I think we take it almost for granted, but basically it is a basis of all our ability to communicate that we largely see the world in a similar manner. It is so obvious that we don't consider it even or contemplate it, except when we are actually confronted with exceptional cases. For example, this very famous picture in which people don't agree on how it appears. But I think it is stimulating these exceptional cases, and of course all the subtle philosophical debates related to it are interesting, particularly, especially because there are exceptions, because in the general domain, we fully agree when we see images on how they look like, we can cooperate, we can communicate, so we can, I think it is fair to say that to a large degree, we share the visual experiences. In other words, we have the similar phenomenal visual experiences as we look around on the same stimuli, despite the fact that we have different brains. So if you kind of consider what you can sort of use this kind of basic shared vision concept experimentally, this makes a very simple prediction that if we compare the coding mechanism for visual perception in one brain compared to another, those coding mechanisms that actually underlie the phenomenal experience of the individuals should be, to a large degree, again, I'm careful and I would say with variation, but to a large degree, they should be correlated to each other. They should be similar to each other. By contrast, coding schemes that you might find in the cortex, but completely change as you move from one individual to another, are probably not underlying the phenomenal experience of vision that is shared among us. So you can, by looking at what remains similar or invariant across individuals versus those mechanisms that change across the individuals, you can get an idea which are the coding mechanisms that are important for phenomenal experience. So this is the rationale behind the experiment. And the experiment, again, this time there are a large data set because we needed many comparisons. So we had 3 sets of patients looking again at images of faces, tools, textures, buildings, and so forth, with slight variation between them. I will not get into it. And again, we collected data from many places in the brain. But in this presentation, I'll mention only high order visual cortex. These are ventral contacts. You see them here. And we collected them, as I said, in many individual patients. And now what Ofer did is simply compared what neural codes, as we compare one brain to another across all the possible pairs, which one remain invariant and which one are changing from one brain to another. Now, the first simplest kind of invariance you might consider is that simply my brain is a copy of your brain. In other words, if we look at the specific high order visual areas that Ofer looked at, perhaps we could test the possibility, consider the possibility that they have a simply identical organization, namely neurons that are the same place in my visual cortex behave, they have a tuning profile of activation that are identical to a neuron located in the same place, homologous place in your brain. I hope this is clear. This is the very simple assumption that locations that are very similar in anatomical space in one brain are copied and have exactly the same functionality in the other brain. So what you see here is Ofer generated a scatter plot consisting of all the possible pairs between contact between all patients. And what you see here on the x-axis is the distance, the anatomical distance, not between 2 pairs within one cortex, but between one contact in one brain and the homologous point in the other brain of another individual, after normalizing the brains and trying to match them as best as possible. And or what you see on the y-axis is how similar the tuning, the properties of each of these contacts was across in the pairs. And if one cortex was a copy paste of the other, we would expect a very strong and steep correlation between distance and similarity, where contacts that are very similar to each other have very sharp tuning, and contacts that are very far away from each other have very low similarity in tuning. Very strong similarity in tuning, and here very low. And you see some kind of slight drift, indeed showing that over long distances, there is some similarity in functionality between one brain and another. But if you look within one specific location, you see that there is a major scatter. The same distance between different contacts can be either very, very close or very far away in terms of tuning properties, meaning over long distances, and that we know, of course, from fMRI studies and so forth, over long distances, different brains are alike, but within local high-resolution anatomical distances, there is a huge scatter, and they are not alike at all. The next option is again going back to our population vector. Could it be that the same barcode for Bruce Willis in my brain is similar to your barcode in your brain? And the way Ofer tested that, he first of all generated the population vector in one brain. Then in the other brain, he permuted the orders of the contacts. So we are not maintaining anymore matching in anatomical arrangement. You are allowed to change the anatomical arrangement as much as you want. You can see that here the contacts are very different, but you are permuting it in an attempt to maximize the similarity between the population vector in one brain and the population vector in another brain. You do it for all possible images, for all possible matches among different brains. And the results for this matching are here. I show it here only for the face categories. In other words, we did this relational structures analysis, only a population vector analysis, only for in the face domain. And very, very important is that you do the permutation. In other words, you try to match as best as you can the population vector in one brain with another on a subset of the images, about 2/3 of the images that you use as a sort of training, in other words, to find the optimal match. But the testing is done on a separate set of images. This is very important, that were not part of the optimization process. But you are using the arrangement, the matching of the contacts that you did in the training, you are using in the testing, and you ask yourself, how correlated are the population vectors across all the images that I showed the patient? The patient, of course, saw exactly the same images. You see here the 3 sets of images, how correlated they are. And the answer is, there is some correlation. It is very, very weak, but there is some positive correlation, some marginal positive correlation across different brains. So population vector, to some extent, but very weakly, is invariant across brains. Now, Ofer turned to relational structures. And how do you match relational structures? You're not permuting the individual neurons. What you are doing is essentially you are rotating. Here I show it, of course, in a caricature, only 2 neurons. You are rotating the vector space, the dimension of the vector space in one patient in order to match as best as possible the angles in another patient, and you again do it for all possible images, all possible rotations. You're trying to find what is the optimal rotation of one dimension, the one set of dimension relative to the other patient that you are trying to match. You find the best optimal match, and then you calculate how good is the match, what is the correlation. And you see the result of this manipulation. Again, you do this rotation or matching of the relational structures on a training set, and test it on a separate images that were not part of the training set. And you see a really robust and striking increase in the correlation across different brains as you move from the population vector domain to the relational structure domain. And the important thing is not only that in the training you get a major increase, but that you get it also in the test. When you do the same kind of rotation on a totally separate set of images. And this was not only for faces. You see here the results done separately for each category, separately for places, tools, textures, and so forth. In all of them, there is a major jump as you move from the population vector to the relational coding. The blue bars are checking control of even a higher dimensional model. I will not get into it. If you're interested, we can discuss it in the discussion session. So the conclusion is that if we look at different brains and you ask what remains similar and what you might say evolution didn't bother to remain similar, what remains similar across brains is the relational structure and not the population vector. The population vector, just like I said, can change completely, can be very different, but the relational structure may be maintained. Now, this kind of result allowed the author to ask a further question, interesting question, which is actually not resolved. And this is, what are the boundaries of a relational structure? You might consider that relational structures are containing only particular category of visual images. For example, there will be a separate relational structure for faces, another one for building, another one for tools, and so forth. Or you might consider maybe relational structures are more inclusive, and they might contain also distances, for example, between animals and faces. And one way to address this question is to ask, okay, so now let's see what remains invariant, what remains the same as we move from one brain to another. Are these pure relational structures that remain invariant, or maybe larger ones where you can include additional categories and still the structure will remain invariant? To put it another way, is the rotation from one brain relative to another confined to a specific relational distance within a category, within the domain of faces or animals, or is the relational structure rotated the same way also when looking at distances between categories? So this is what author compared. They compared the invariance when you do this matching separately for different categories, and the invariance across individuals when you are mixing categories. And the result is clear-cut. You get a much better match invariance across individuals when you are using separate categories, and this is degraded when you are mixing different categories. And if you think about it, it makes some phenomenal sense also. It's very easy to say whether faces are similar or not. It's much more difficult to say whether a house and a face are more similar or not. I mean, it's not really clear how you even answer this question. So this somehow maybe provide a suggestion why is this the case. Okay, so finally, I would like to be a bit more speculative and try to sort of discuss relational structure in terms of neurons more deeply. And basically, I will start by posing a question that I'm keep being bombarded with consistently, and it might occur to you also. And this is the question. I keep talking about seeing a face as an appearance of a face as a result of the relation of this face to other faces. But when I show you the face of Barack Obama, I don't in parallel show you all the other possible faces for you to make a judgment and then decide that this is Barack Obama. You experience Barack Obama, and I will argue you experience it because of relationship, but I never actually showed you this relationship. I'd never shown you the other images. So how come I argue that you see Barack Obama because of the relationship, when in fact I never allowed you to experience this relationship directly? And this is a deep question that, to be frank, I don't have the answer, but I would like to propose a suggestion that is compatible with some results that we have and actually ties it to another issue that you might be thinking all along, what this whole thing has to do with consciousness. It might be that you have these relational structures also when you're totally unconscious, and why am I bothering you with focusing so much on that? And I think what I'll discuss now might try to touch on it. Again, speculative. I will not say that I have the definite answer here. So basically, what does it mean, if you think about it neuronally, what does it mean that I'm talking about a relational structure? What I mean is basically coded information, some specific information or structured information that is coded in a particular local region in the brain. And we know very well how information is coded in neurons. This is, an old line discovery and finding and so forth. information is coded in neuron by the synaptic weights between the neurons in the network. So very simply, the idea of a relational structure is simply the idea that there is a unique pattern of synaptic weights within a local region, and we expose this synaptic structure, or we learn about it as scientists. We can, of course, don't have microscope and technique to go down into the synaptic structure and study each synapse, how strong it is and how the different neurons are related to each other. What we do is we present these structures with images, measure the representational dissimilarity distance, but this is basically a way for us scientists to expose the underlying structure, synaptic structure, that is within the local region that we call the relational structure. And what I would like to propose is that I think a neglected aspect of the human cerebral cortex is actually a perfect mechanism that could underlie this emergent of or this generation of relational structure. And this anatomical feature is the local lateral connection that connect neurons, neighboring neurons within a cortical region. This is people usually don't realize that, but this is one of the most intense matrix of connections that are known in the cortex. They're actually the defining character of the cerebral cortex. We typically think about neuronal connections as a long distance communication line connecting you from one area to another, but this is not the case. The anatomy tells us that within each cortical region, we have a massive halo of connections, and I showed you some old study that we did where we mapped this intrinsic connection. This is, I think, 0.5 millimeter in size. So these are very local halos of connection. What you see here is the connections emanated from where the arrow is in the group of neurons and how intensely they communicate to their neighboring neurons. Now, this is a random injection. If I injected the same tracer here, this is by the way done in primate cortex. If I injected the same tracer here, I'll see exactly the same halo. So you can immediately understand that we are talking about here a massive recurrent matrix of connectivity. Now you can already see that what you see here are, for example, from different visual areas in the primate. And you can see that each area has its unique signature of these intense lateral connections that are determined. They change in length, they change in intensity, in density, in spread, and so forth. So what I would like to propose, that what you are looking at here is basically the substrate of the relational structure. And we simply expose this structure for study by doing relational measurements. In other words, we expose this network to different images. We record the activation that emerges due to the interactions within these networks. And then we display all these distances. We call it the relational structure. But this is the relational structure. This is just a sort of a way to expose the relational structure. Now, how this might be related to consciousness? Now, a finding from my group and replicated by others is a very interesting dynamics that you observe when you contrast the classic threshold experiments. In our case, it was a backward masking experiment. I will not get into detail, but the idea is very simply, you present images very briefly and you mask them with a high density noise. And then you can get a threshold situation where the same brief presentation of an image, in this case, faces again, is being reported as being seen by the subjects. And they say, I see the face. Or with a slight, other times, exactly the same stimulus, they will completely fail and say, no, I don't see the images, I just see noise. And you record with these intracranial recordings during this work done by Lior Fish And you record and you simply contrast the dynamics of the activation when the patient saw the images and with the same stimulus when they didn't see the images. These recordings were again done in this phase contact, phase selective ventral contact. And you see two interesting signatures of the visibility or the reported visibility of the images. One of them is a massive amplification of the signal. I remind you, the signal is the input is the same, but when they see it, you see a big amplification of the response as opposed to when they fail to see it. And the other one is that the response becomes more sustained. These two signatures of a nonlinear increase in activations and sustained response, I remind you, the stimulus ended within about, if I recall right, 20 milliseconds. The response lasts for about 600 milliseconds after the stimulus is gone and masked by mask, and it keeps persist. This is a classical signature of recurrent activity, what we call local ignition. The metaphor is exactly that, an ignition of a neuronal fire. You increase beyond a certain threshold, the neurons, because of this very, very dense lateral connection that I showed you, will activate each other and then will create this neuronal fire that is both amplified in amplitude and sustained. Again, we need additional studies to verify this model, but basically the idea is that whenever you see an image, there is a massive burst of recurrent activity that is generated locally within these local structures. Now, what does it mean that you have a recurrent activity of the neurons within the local structure? Basically, the neurons are sort of expressing or testing, you might say, the synaptic weights between them. They activate each other, and in this way, they sort of manifest or exhibit the nature of the synaptic connectivity. So the response, the conscious response that we call a pattern of activation, is in fact a result of exploring and activating the relational structure, and as a result, getting the activation pattern. It's not that you need to do the relational comparison in order to get the pattern. You get the pattern because of the relational comparisons that under certain inputs, thinking, recall, will generate as a burst of ignition the specific content that you are seeing. So what I would like to argue is that when you have a relational structure emerging, that means you are also have the local ignition, and this ties the suggestion that I would like to make, that relational structure is not only a coding for information, it is coding for phenomenal information. And when you are not conscious of the phenomenal experience, you simply don't have a relational structure that is being expressed. It remains dormant within the synaptic contacts within an area. So I'll finish here. So basically, what did I try to tell you or convince you today? First of all, I would like to argue that a relational coding, namely the matrix of similarities and distances between different exemplars within a phenomenal category, is the code, is a central code in determining or defining or coding for this content. I showed you that in line with this concept, we do find a correspondence between neuronal pattern similarities and distances and the perceptual similarities when we're looking at the ventral phase related regions. We find the relational structures to remain invariant across individuals, despite the fact that the actual population vectors may change. The brain seems to, across individuals, seem to maintain their similarity of the relational structure. We find that we can decipher the functional specialization of different cortical areas through their correlation to different DNNs that perform different objective functions. In this particular case, text versus vision. And we find that frontal relational structures appear to be text related. And what I would like to propose is that you find relational structures only when you are conscious of the experience, of the relevant experience. And the way relational structures are actually expressed or sort of displayed during phenomenal experience is through this local recurrent burst of local recurrent activity that in fact uses the synaptic strength or weights in order to generate the pattern that you see. So overall, I would like to argue that the results support the hypothesis. The local relational coding is central in underlying conscious content. So thank you for listening, and I would like to acknowledge the contributors to this work. From my lab at the Weizmann, Rotem Brodeit-Weir, Itzhak Norman and Shani Grossman played important roles. From Reichman University, Ofer Lippmann, Doron Friedman, and Yaakov Helor were studying the relation across brains. And from Tel Aviv University, the lab of Galit Yuvel, Adva Shoham, together with Itai Yaron, explored this idea of relationship to deep convolutional network. Thank you.
Fantastic. Thank you, Rafi.